Welcome to Crafty Medicos. In this session, we are going to learn about the reproductive system histology, and basically in this video we are concerned with the histology of the male reproductive system. So the male reproductive system contains the several organs, for example the testes, and within the testes there are the several tubules which are called the seminiferous tubules, and then there are the glands and the ducts that are associated with the male reproductive system. We will learn about the histology of each and every structure and every segment in detail in this video. So first, testis is considered as an endocrine as well as an exocrine gland. It's considered an endocrine gland because it produces the various hormones, for example testosterone, as an exocrine gland because with the help of a duct that is called the vas deferens, the sperms that are produced inside the testes are transmitted through that duct. So it's considered our ducted gland, that is the exocrine gland. So let's learn the histological structure of the testis. So the testis contains a capsule that is the dense irregular connective tissue and is called the tunica albuginea. This tunica albuginea is thickened posteriorly, which is to form the mediastinum testis. Now from this mediastinum testis various fibrous septa arise, and this mediastinum testis is actually the entry and the exit point for several blood vessels and the lymphatic vessels. Now the beneath the tunica albuginea there is a loose connective tissue which contains the blood vessels, and it's called the tunica vestibulosa. As I already mentioned that there are the fibrous septa that arise from the mediastinum testis, and these septa then penetrate the organ from the fibrous side and divide it into 250 pyramidal compartments which are called the testicular lobules. Now these testicular lobules, each of these testicular lobule contain 1 to 4 highly convoluted seminiferous tubule, and between the seminiferous tubule there is a sparse connective tissue which contain the various endocrine interstitial cells that are called the Leydig cells, and this sparse connective tissue is called the intertubular connective tissue. This is the outer covering of the testis, that is the tunica albuginea, and it is thickened posteriorly where it forms the mediastinum of the testis. Now from this mediastinum of this testis you can see various septa that arise from this testis and divide it into the several lobules. Then you can also see the highly convoluted tubules within each lobule that are called the seminiferous tubules. Let's now discuss about the seminiferous tubules. These are the highly coiled and convoluted tubules, and they end as a free blind and near the apex of the lobule, and they continues into a short and narrow tubulus rectus, also called the straight tubule. So here you can see that the seminiferous tubules end as the straight tubules, and then these straight tubules become continuous with the rete testes that are located in the mediastinum of the testis, and then these rete testes continue with the efferent ductules, and these efferent ductules then open into the duct of the epididymis in the head region of the epididymis. So from this diagram you can get a clear idea that how the sperms that are formed within the seminiferous tubules of each of these lobules are transported into the epididymis. So here is the epididymis, and here you know the network of the channels. For example, first of all we have the tubulus rectus or the straight tubules, then these lead to the rete testis, and the rete testis then continue with the efferent ductules, and then efferent ductules become continuous with the duct of the epididymis. Let's now talk about the seminiferous epithelium. So this seminiferous epithelium is also called a germinal epithelium or a spermatogenic epithelium. It is actually a modified stratified cuboidal epithelium, and it rests on the basement membrane. Outer to this basement membrane there is a thin layer of the fibrous connective tissue which contains the smooth muscle cells which are called the myoid cells. So what's the purpose of these myoid cells? That the contraction of myoid cells help to push the spermatozoa through the seminiferous tubule into the tubuli recti. This is the histological diagram showing the seminiferous tubules. So you can appreciate there is a basement membrane, and the epithelial cells of these seminiferous tubules are resting on this basement membrane. Then outer to basement membrane there is a connective tissue, and this connective tissue contains the cells which are called the myoid cells, which have the ability of the contraction, and the function of which we have already discussed. Now we will discuss about the cells that are present in the spermatogenic or the seminiferous epithelium. First of all we have the spermatogenic cells, and these spermatogenic cells lie between the Sertoli cells and form several layers of cells which occupy a space between basal lamina and lumen. Before puberty, only primitive germ cells called the spermatogonia are present. However, at puberty, the process of the spermatogenesis begins under the influence of the testosterone. Thus, in an adult male, different stages of spermatogonia can be distinguished based on the changes in the shape and the staining properties of their nuclei near the basal lamina of the epithelium. We'll see the different types of the spermatogenic cells. First of all, we have the type A dark cells. These type A dark cells have the oval dark staining nuclei and their nucleolus is close to the nuclear membrane. These type A dark cells are the reserve cells and they divide to maintain their own number and also give rise to the type A pale cells. These type A pale cells then divide to form the type A pale cells to reserve their own number and also give rise to the type B pale cells. These type B pale cells have the spherical nuclei and they show the dark staining clumps of the chromatin. Their nucleolus is situated in center of the nucleus. These type B cells then divide to form the primary spermatocytes and these primary spermatocytes then undergo the meiosis 1 to form the secondary spermatocytes, which are the smaller cells, and then secondary spermatocytes then divide to form the spermatids and spermatids differentiate to form the spermatozoa or the mature sperms. Now after doing the spermatogenic cells, which are the first type of the seminiferous epithelial cells, the second type of the epithelial cells are the Sertoli cells, which are the tall columnar sporting epithelial cells and they extend from the basal lamina to the lumen. And due to their close association with the spermatogenic cells, their lateral margins are irregular and can't be distinguished. Their nucleus is large, oval and vesicular and is located in the basal portion of the cells and it contains a prominent dark staining nucleolus. Their cytoplasm contains large number of the mitochondria, large number of Golgi apparatus and extensive SCR and some profiles of RER and lysosomes also. The compartment, the lateral walls are irregular and there are the tight junctions between the cytoplasmic extensions of the Sertoli cells. Due to these tight junctions, the epithelium is divided into two compartments, the basal compartment and the adluminal compartment. In the basal compartment, there are the pre-meiotic germ cells, for example, the spermatogonia and the early primary spermatocytes. Now, as the primary spermatocytes mature, the occluding junctions disassemble and primary spermatocytes move into the adluminal compartment. The spermatids are closely applied to the surface of the Sertoli cells. Luminal compartment contain the meiotic population of developing germ cells which include the dividing primary spermatocytes, secondary spermatocytes, spermatids and developing spermatozoa. So mainly the cells whose nuclei contains the haploid number of chromosomes are included in the adluminal compartment. And here is the diagram showing the basal compartment as well as the adluminal compartment. So you can see in the basal compartment there are the primary spermatocytes and the other pre-meiotic cells, whereas in the adluminal compartment there are the spermatids, differentiating spermatids which then differentiate to form the sperms. Now let's see the functions of the Sertoli cells. So these Sertoli cells have the functions of support and nutrition. Because the germ cells are isolated from blood, so the oxygen and all the nutrients are provided from the intertubular connective tissue through the cytoplasm of the Sertoli cells to the spermatogenic cells in the adluminal compartment. And these Sertoli cells are also involved in the protection of the germ cells. Germ cells contain the haploid chromosomes and therefore are protected from the autoimmune attack by creating the blood-testis barrier. Then the secretory functions include the synthesis of many substances such as the testicular fluid, the androgen binding proteins, the inhibin, the Mullerian inhibiting factor, which have the various functions. For example, the testicular fluid serve as the transport medium for the newly shed immature non-motile spermatozoa. Androgen binding protein concentrate the testosterone in the adluminal compartment, whereas the inhibin is released into the blood in endocrine manner and it is involved in the feedback loop which inhibits the FSH. Mullerian inhibiting factors suppress the development of the paramesonephric ducts, which are the main ducts in case of the development of the female genital system. Then there comes the phagocytic functions of the Sertoli cells. The Sertoli cells act as the scavengers and phagocytize the excess cytoplasm of the spermatids during the spermiogenesis, and the residual bodies are engulfed by the Sertoli cells and broken down and digested by the lysosomal system of these cells.